that is very interesting, I think. And we can do a lot in this field to address the current challenges in the field of cancer imaging and cancer therapy. So please let me get into the talk and give you a brief introduction to the cancer nanotechnology or cancer theranostic. So as many of you know better than I do, the current methods for cancer therapy include in surgery, radiation therapy, chemotherapy. These three are the main therapeutic ways to treat the cancer. But there are some other methods to treat cancer, like immunotherapy, hormone therapy, targeted therapy, or stem cell therapy that I have been recently doing some research in the field of combination of cancer's nanotechnology and stem cell therapy to use the stem cells for nanoparticle delivery to cancer cells. I will talk about this maybe at the end of this talk, but let's get back into the current conventional methods for cancer therapy that are surgery, radiation therapy, and chemotherapy. So as you know, surgery is invasive method, and also radiation therapy and chemotherapy. These two methods are severe side effects because they are not selective method to only destroy the cancer cells. So collateral normal cells also get damaged, and we have to think about modification of these non-selective methods. So nanoparticles can be promising candidates to address this non-selectivity, and I'm sure you know pretty much better than I do how many types of nanoparticle we can do at this time, like lipid-based nanocarriers, polymer-based nanoparticles, like viral nanoparticles, and also maybe we can categorize the nanoparticles that we can use in the field of medicine or specifically in the field of cancer therapy or imaging into organic and inorganic nanoparticle. That my talk today will be in the field of metallic nanoparticle, and also oxide nanoparticle like iron oxide nanoparticle, and also plasmonic nanoparticles like gold, as the example of the talk I have today. So if we combine therapeutic effects and diagnostic effects, then we can say we have theranostic nanoparticle, which can be able to address both therapeutic applications and diagnostic applications. So theranostic stands for therapeutic and diagnostic. And in this case, we have a nanostructure that can be used for imaging and also for therapy. By imaging, I mean we have lots of methods that can be used for cancer detection, like MRI, CT, PET, ultrasound, or sonography, photoacoustic imaging, and the recent one that is kind of new molecular imaging method that is called as the MPI, that we started this technique at Hopkins last year, and it stands for magnetic particle imaging, that can be specifically image the presence of particles inside the body. So with this imaging method, we can guide the therapy of cancer, and also we can combine the therapy with imaging so that with the guidance of imaging, we can do drug delivery or gene delivery, or we can do hyperthermia, and through thermal ablation or thermal destruction, we can kill the cancer cells, or sometimes we can do photodynamic therapy, in which uh, we use a photodynamic agent, and through some chemical reaction that is induced by lasers, appropriate laser, we can do photodynamic therapy. And all these processes can be guided by imaging, and the probe that will help us in this regard is the nanoparticle that I am going to talk about that. So to do image guided therapy using this theranostic nanoparticle, I'm going to talk about the specific type of theranostic nanoparticle that I have been working on that for the past 7 years. And it is made of SPO, or super paramagnetic iron oxide nanoparticle, and also AUMP, or gold nanoparticle. But we have lots of options to do and to make this specific nanostructure. So as you can guess, my talk would have 3 parts. In the first part, I'm gonna talk about SPO and some characteristics and applications that this kind of nanoparticle may have. And in the second part, I'm gonna talk about gold nanoparticle as a plasmonic nanoparticle that we have lots of options to do cancer treatment. And finally, I'm going to talk about combination of these 2 kinds of nanoparticles, and how we can guide the cancer therapy using this multifunctional nanoparticle. So in the first part, I'm going to talk about SPO nanoparticle and the applications that this kind of nanoparticle has. So the first and most popular and most well-known application that SPO nanoparticle has is the application in MRI, or magnetic resonance imaging. As many of you know, we can use these nanoparticles for weighting of the MR images. Like we can do T1 or T2 weighted MR imaging, and it depends on the size of the nanoparticles that we are using. If we use super small nanoparticles, we can do T1 weighted MRI. If we use larger and bigger nanoparticles, so we can get T2 weighted MR imaging. So depending on the application that we have in mind, we can modify and we can play with this characteristics that this kind of nanoparticle has. So we have lots of opportunity, and I have to mention here one. I think this is the most important thing that I'm gonna talk about MRI and then MPI. So when we are talking about imaging and probe-based imaging, sometimes we call the probe as the contrast agent, and sometimes we call the probe as the tracer. When we have a method in which the tissue of the human body has a signal like MRI, like CT, then we have to call the probe as the contrast agent, because like here, we can see all the parts of the body and other tissues, but using the probe that we are developing, we are going to increase the contrast so that the specific region of interest get darker or get brighter, right? It depends on the weighting of the MRI. But sometimes, like MPI, that I'm going to talk about that later on, there is no background and there is no source for the signal from the tissue. So when we don't have any background and we only can see the tracer, the nanoparticle, then we call it as the tracer. So MRI and the use of nanoparticle we have to say we are talking about contrast agent, not tracer. So the probe is called here contrast agent. And with this, I'm going to mention that we have lots of options to make different types of magnetic nanoparticles, spherical, star-shaped, different type that you are pretty much better than me, and you know how to make them and how to optimize the characteristic of these particles. And we have nothing about that, but we use these nanoparticles to get the contrast enhanced, or maybe we can make the region of interest, like a tumor, brighter or darker to specify where it is located. So let me get into MPI, or magnetic particle imaging. The concept is somehow the same as MRI, but the physics of MPI and MRI are completely different. As I said, in MRI, we only look at hydrogen distributed across the body. And using the magnetic nanoparticle, we shorten the relaxation time of hydrogen. So MRI is the machine to look at hydrogen, but MPI is a machine to look at the magnetic nanoparticle. So this means that because we don't have any magnetic nanoparticle as the background in our body, so let's say this is MPI of a mouse, okay? So wherever you don't have any magnetic nanoparticle, like here, you would get no signal here. But when you have hotspot here, this means that you have accumulation of the magnetic nanoparticle, like let's say in the brain, or here, let's say in the liver, they are accumulating after injection. So with this, I'm gonna emphasize that MPI is a tracer-based imaging, but MRI is a contrast-based imaging. So what are the benefits of MPI over MRI? I can address this question like this. With MPI, we have high sensitivity, much higher than MRI. The sensitivity is depending on the parameters that we do imaging. Sensitivity in MPI is much higher than MRI, and the benefit of MPI over MRI is that we can do absolute quantification. As you know, with MRI we cannot do quantification because of magnetic susceptibility effect. When we have high concentration of magnetic nanoparticles, then we get some kind of saturation and we cannot do any quantification. But MPI is a good method to do quantification and estimate how many nanoparticles are accumulating here in the brain or in the liver or lung or somewhere else. So MPI can be considered as the absolute quantification quantification method, and it is independent of depth. And also we can do longitudinal tracer detection non-invasively, and it's kind of similar to what we do in nuclear medicine, like PET scan. But the benefit of MPI is that we are using cold agent. In PET, we use hot agent like radioactive material, but here we only use magnetic nanoparticles that are cold, and there is no radiation activity and no radioactivity. So this is the main benefit of MPI over PET. But it's not all rosy, and there are some limitations with MPI. The main problem that MPI has is that there is no anatomical information with this imaging modality. So we always have to do registration. Like here, we use CT or MRI to do to get anatomical information, but it's easy. We can do co-registration of MPI dataset on CT dataset, and it's not a big deal. But the other problem that it has is the poor resolution that this technique has. So I know engineers are working on this technique to increase and improve the spatial resolution of this technique. So after mentioning the imaging potential and capabilities of magnetic nanoparticle, I'm going to talk about therapeutic application that this nanoparticle has. The most important thing that I should say here is the application of magnetic nanoparticle in MFH, or magnetic fluid hyperthermia. That is the most common hyperthermia method that everybody knows about that. The idea is to load the cancer cells with magnetic nanoparticle and then oscillate them using an oscillating magnetic field and get the temperature of the cancer cells increased up to 45 degree, and then we can kill the cancer cells through denaturation of the proteins, and we can destroy the cancer cells and finally the tumor. But the limitation is again non-selectivity, because magnetic nanoparticle can be internalized by any kind of cells. So we should do some kind of targeting, and this is the main challenge that MFH, or magnetic fluid hyperthermia, has. For example, here, let's say this patient has a tumor in the brain, and we injected magnetic nanoparticle, and nanoparticle are distributing in tumor and also in normal brain tissue. So if we expose the head of this patient with radio frequency wave to make the nanoparticle oscillating, so there is high chance to damage the normal tissue in the brain, and side effect will be happening. And so off-target accumulation for non-targeted nanoparticle always occurs, right? So we need to target the nanoparticles to tumor site. First solution is to target magnetic nanoparticle towards cancer cells using molecular targeting. By molecular targeting, I mean we can conjugate the nanoparticles to some targeting agent like vitamins, like antibodies, something like that, which can be which can carry the nanoparticles. Carry is not very good term here. Actually, the nanoparticle can be selectively internalized by cancer cells, because there are some receptor on the cell membrane, I mean cancer cell membrane, and the chance of internalization of targeted nanoparticle is much higher than normal cells. So with this, we can do molecular targeting, but unfortunately, there are some negative report on molecular targeting methods. Let's say folic acid is very good moiety or ligand to target the nanoparticles to our cancer cells, and we did this review article and published in Nano Today, and we address the challenges that folate targeting has. Folate targeting is very good way to target the nanoparticles toward cancer cells, but unfortunately, there are some reports that there is non-specific accumulation of the targeted nanoparticle in kidneys, in liver and spleen. So, for example, here, if you look at folate targeted nanoparticle and non-targeted nanoparticle, you can see accumulation of targeted nanoparticle in kidneys, while non-targeted nanoparticle, that you cannot see any accumulation in kidney. So this is the most important challenge that molecular targeting has. So the other way to address non-selectivity of MFH is to get help from engineers. And engineer can help us to develop some methods to first see the magnetic nanoparticle, and then to do thermometry, and then we can specifically do localized hyperthermia, and with the guidance of imaging that I'm going to talk about that maybe by the end of this talk. So the idea of engineering solution is to first see the nanoparticle and then hit the nanoparticle. As I mentioned in a couple of previous slides, we have two options to see the magnetic nanoparticles. The first one is MRI, the second one is MPI. But for MRI, as I mentioned, that with MRI we cannot do any quantification, and at high concentration this technique is disabled to do quantification. So we need to think about other methods, and MPI is very useful here because we can do quantification with MPI even at much higher concentration, and there is no saturation with this technique. And as you know, for hyperthermia, we need high concentration of nanoparticles. So the combination of MPI and magnetic hyperthermia is one of the, it seems that it's one of the most promising way to first see the nanoparticle and then hit the nanoparticle. And here there is a paper, I think there is only one paper in this field published in ACS Nano so far. The researcher here, they injected magnetic nanoparticle. Here is tumor and here is liver, and it's kind of off-target accumulation of nanoparticles in liver. So if we expose this animal generally, so both liver and tumors will be destroyed, right? So, but the only target should be tumor, not liver. With this technique, when we specify the hotspot in liver and tumor, then we can localize the magnetic field for treatment only on tumor. And using the localized magnetic field on the tumor, they confirm that liver is a cold spot, but tumor is hot spot, and they can destroy the tumor site more specifically and selectively. So this is kind of magnetic nanoparticle mediated MPI and hyperthermia. And here at Hopkins, we have the machine for MPI and also for hyperthermia. And the benefit of combination of these two machines is that, let's say here we have an animal, this is the mouse, and we have injected the magnetic nanoparticles, and we can see the particles here. Let's say we only need to expose this part or this part, then we did the guidance of MPI, we can focus the hyper radiation or hyper field on only on this hotspot, and all these hotspot, other hotspot can be excluded from the treatment field. And this is the one of the project that we are doing at Hopkins. And second part of this talk is about gold nanoparticle. And as you pretty much know better than I do, plasmonic nanoparticles can be excited if we use proper laser wavelength, and if we activate them with proper laser, because of the plasmonic or SPR that you know. I'm sure you know this effect better than me. its surface plasmon resonance, or SPR, that is vibration of conduction electrons, and it depends on the shape on the particles. If we have a spherical gold nanoparticle, we have one SPR peak, and if we have like rod shaped nanoparticle, we have two peaks for SPR, and for transversal and longitudinal oscillation, we can get two peaks. And if we use the proper and matching frequency for the laser, or wavelengths for the laser that is matched with this peak, then we can get the nanoparticles heated, and we can use this heat for hyperthermia or photothermal ablation to kill the cancer cells. And with this, I'm going to talk about the different and diverse options that we have in the field of plasmonic nanoparticle, and specifically in the field of gold nanoparticle. And here you can see that there is diversity in plasmonic nanoparticle. We can make them spherical, rod shape, star shape. We can make assemblies like aggregates, two-dimensional, three-dimensional. There are lots of options that we have, and every kind of this nanoparticle has a specific advantage and disadvantage that is that should be discussed in another talk. But here I'm going to talk about the application of these nanoparticles in cancer imaging and cancer therapy. Um, we have published this review article, which is kind of comprehensive review article. And in this paper, we reviewed all the options that we can get with gold nanoparticle in the field of cancer therapy. And we can do radiotherapy. And if we target the gold nanoparticle in cancer cells, then using the radiation, I know there is active radiobiology group in your university and in your institute, so maybe they are interested in this field. We can use gold nanoparticle and targeted gold nanoparticle in the field of cancer radiotherapy, and also we can use gold nanoparticle to target the chemotherapy chemotherapeutics toward cancer cells. We can use them in immunotherapy, photothermal therapy, and in this article, we have reviewed all the options that we have with gold nanoparticle. And actually, this is kind of game changer. Conventional method of hyperthermia, if we use laser, ultrasound, alternative magnetic field, or radio frequency, this kind of hyperthermia is out inside hyperthermia. This means that we have a source of energy outside of the body. So if the target or tumor is here, we have hot spot in the field of and in the path of the laser or ultrasound. So it's kind of out inside hyperthermia. But if we localize the nanoparticles in the tumor, like here, and we decrease the energy of laser or ultrasound, we can change the paradigm and change the game, and we can do inside out hyperthermia. That means that we can spare the tissues in the path of the waves, but we can increase the temperature of this target or the tumor. And more specifically and selectively, we can do cancer treatment using these nanoparticles. But there is a paradigm shift recently in the field of cancer theranostic or cancer nanotheranostics, that is combination therapy or trimodal cancer therapy. And we reviewed that in our paper that I mentioned in 2 or 3 slides. It was in my previous slides. So trimodal cancer therapy is that to take advantages of the gold nanoparticle in all radiation therapy, and photothermal therapy, and also chemotherapy. And when we conjugate and combine all these potentials, we can decrease the session of therapy. Let's say for conventional radiotherapy, maybe we need to treat the patient at least 20 sessions. So we can decrease the sessions, we can increase the quality of life, and quality of treatment, therapeutic ratio, if we combine and take advantage of synergistic effects of these different kinds of therapy using nanoparticles. And finally, the last part of my talk today is to combine gold and iron oxide, and we make one single nano platform that contains both part of gold and magnetic nanoparticle. Uh, and using this nanoparticle, we can address different challenges that we have with image-guided therapy, because in this nano platform we have component for imaging, and we can guide the imaging using magnetic nanoparticle, and we can use the gold nanoparticle for radiation therapy or photothermal therapy, and we can do image-guided therapy. Also, a gold nanoparticle by itself can be used for imaging like photoacoustic imaging or CT imaging. So we have lots of options with these nanoparticles to do imaging, including MRI, MPI, photoacoustic imaging, magnetophotoacoustic imaging, or CT scan. And also for therapy, we have options for radiotherapy, photothermal therapy, and also because we have magnetic nanoparticle, we still have the chance to do magnetic fluid hyperthermia. At the same time, we can do dosimetry, we can do distribution study, and we can monitor the therapeutic and therapy session and how it is going on. In this paper, uh, that is published in scientific report, actually they did a proof of concept study that using the magnetoplasmonic nanostar, they could do MRI and photoacoustic imaging and MPI, some kind of multimodal imaging, and as I said, at the same time they can use the same nano platform for treatment. And also recently we published another paper in the field of image-guided cancer therapy. If you want, you can look at this paper. Actually, after injection of gold-coated iron oxide nanoparticle, we can get MRI. Here is the tumor, and here we can see that the nanoparticles are accumulating in the tumor. Using the MATLAB and COMSOL multiphysics software, we can do simulation, and we can optimize the treatment session, and then we verify the simulation, and then we follow up the animals after treatment, and we saw that this kind of simulation guided photothermal therapy using MRI and also traceable MRI traceable iron oxide nanoparticles, we can guide the therapeutic session. And finally, I'm going to talk about the project that we are doing here in the field of MPI guided hyperthermia. As I said, conventional magnetic fluid hyperthermia has a side effect because there is a high chance to irradiate the normal part of the brain, and we can destroy the normal part of the brain. But if we do MRI or CT MPI guided hyperthermia, then we can localize the therapy field or magnetic hyperthermia only to this region of interest, and only irradiate this part of the brain and selectively damage the tumor in the brain. And using this nano platform, that is magnetoplasmonic nano platform, all these features can be possible. So in conclusion, I'm going to say that the current challenge that we have is to do non-invasive imaging and also treatment of cancer. And one of the promising methods is to use image-based therapy using multifunctional nanoparticles like gold-coated iron oxide nanoparticles. And this kind of nanoparticle is promising agent for both therapy and imaging and to be used in image-guided therapy. And also because of multifunctionality that this kind of nanoparticle has, we can do some kind of paradigm shift from monotherapy to combination therapy. And as I said, the main benefit of this nanoparticle is that we can use this nanoparticle in image-guided cancer therapy. Thank you so much for your kind attention. I would be more than happy to take your question.
Okay, thanks. Thanks, Ali, for your nice and comprehensive talk.
You're welcome.
Yeah, so now it's time for question. If you have any question, you can write it up in the chat box or raise your hand. Then I will give you access to speak and ask your question. I think Rosie has a question. Yes.
Hi, Ali. Thank you so much for your nice presentation. I'm a computational material scientist, so I'm wondering if you or the research group in this field have done any computational modeling or even experimentally. I want to know how do you find out the best shape of nanoparticle or how is the best size of the nanoparticle for this topic, and also how do you determine the best ratio of iron to gold atoms, for example, because I know that the plasmon spectrum of gold nanoparticles is very dependent on the shape and on the size of the nanoparticle. So I'm wondering if any systematic study has been done on determining this parameter, experimentally or modeling wise, you know?
Perfect. It's very good question, I think. What we are doing at Hopkins and also for the past 7 years that I am active in this field, we didn't do any optimization for this. And it's very important question. Sometime we should do this. And, I didn't see any comprehensive study to compare different ratio of gold to iron. Um, I didn't see it yet. Did see it yet. The other thing that you asked is the shape and also physical chemical properties of, let's say, magnetic or plasmonic nanoparticle. There are extensive research that are happening in the world, but I don't have any specific answer to this question to address specifically and cite or refer you to specific review, but I'm sure there are lots of review only on single part of this multifunctional nanoparticle. I mean, there are lots of review on gold nanoparticles or on magnetic nanoparticles, but I could not yet find any comprehensive review in that people reviewed multifunctional nanoparticles like gold and iron oxide in terms of what you did ask. So I think it's one of the room that we can do some research in this field to do optimize to get the parameters optimized for cancer therapy and cancer imaging and to increase the ratio of therapy at the end. But yeah, I think this is very important. Sometime we should do this.
Thank you. I also have another question. So from a therapy point of view, because I wanted to know if there is any a specific wavelength for plasmonic spectrum that you are interested. So do we have any restriction on the wavelength? For example, if you want to use laser.
Yes.
I'm
For photothermal therapy, and whenever we are using laser as the source of energy, yes, there is limitation because uh, for any kind of laser we have effective penetration. So it's better we focus on near infrared region because in this region we have the highest amount of penetration, but it's not that much, the effective penetration is like 5 millimeter. So what we can treat with this kind of therapy method is to treat superficial cancers, or the cancers that are located in a lumen or in a cavity, like in stomach, or like cervix cancer, wherever we can guide the optical fiber, and also we can deliver the laser using optical fiber. But let's say for brain or for liver, I have seen there are lots of research in this field, but I think they are not promising because there is serious limitation to deliver the light effectively. So the best answer to your question is that if you are going to develop some photosensitizer, it's better you focus on near infrared region.
Yeah, thank you so much.
Sure, thank you.
Prof. Maza, the floor is yours. You can ask your question, Prof.
Thank you. Thanks a lot. Thank you, Prof. Shakerizada, for this inspiring talk indeed. My question is that in terms of hypothermia, I would expect that materials which possess high magnetic moment would be ideal. Why the limitation, or more or less within the literature or the research which is carried out on hypothermia, why we are focusing mainly on iron oxides? Why not, for example, cobalt oxides, or oxides which possess a higher magnetic moment?
Right. Actually, there are lots of options that we can do magnetic hyperthermia. As you said, we have cobalt, uh, or manganese, or the other magnetic nanoparticle that we have, and also people are trying to develop specific ferrite or some other kind of magnetic nanoparticles. And I know there are lots of review paper comparing the magnetic properties of these different kind of magnetic nanoparticles, but the main reason, I have no idea why iron oxide is leading nanoparticle at this time, but maybe it's because this nanoparticle and formulation of that, like resolvist or ferrocarbotran or other kind of magnetic nanoparticle, get FDA approval already, and they are using for liver imaging with MRI, and maybe it's much easier to get them translated in clinic. I think this is one of the reasons, but I'm sure there are so many other reasons that I don't know that. Yeah.
Thank you. Thanks a lot.
Sure. And also this is the same for MPI, professor. For MPI, the other problem that we have is whenever you develop a new nanoparticle, it takes a long time to get it translated and get all the permissions that you need to use it in the clinic. And the benefit that we have with MPI is that we can use the same agent that already got approval from FDA to be used for human, but it's not a general rule. I mean, good MRI agent is not necessarily good MPI agent, and it is the same for hyperthermia. Good MRI agent is not necessarily good particle for hyperthermia, right? So if you have multifunctional nanoparticle that can be used for all modalities, then I think you nailed it down.
Thank you.
Sure.
Adris, please proceed with your question. Okay, Adris, your hand is raised for question. If you can hear me, please ask your question.
Okay, I think they can hear me now. Can they hear me now?
Yes, yes, we can hear you very well.
Oh,
Please
sorry, I
ask your
was
question.
actually muted myself. Thank you so much for the fantastic presentation. I have some couples of questions to ask. The first on the list is that did you take cognizance of the biocompatibility properties of the nanoparticles? Because of, I'm looking at what you are using it for theranostic. So that's the first question. And the second question is that do you also take cognizance of the charge of the nanocomposite that you are using for this application? And thirdly is that I'm also looking at the size, because sometimes size plays a major role in the electron migration of the nanoparticles. And I'm also looking at the interaction that takes place between the nanocomposites that you are using for the materials. Are you looking at it from the negative charge, or must it has a positive charge particles, especially when you are blending nanomaterials together? And I'm also looking at it from the fact that the synthetic route that is being used in the preparation of the nanoparticles, looking at it from the angle of cost angle, because we know we can prepare different nanoparticles through hydrothermal, co-precipitation, and all the likes. So those are the questions that is actually coming to my mind. Thank you, sir.
Sure. Thank you for all the questions that you asked. I'm going to start with biocompatibility. As you mentioned, this is very important feature that when we develop a new nanoparticle, we should look at that. Actually, we in one research, we quoted gold iron oxide nanoparticle or magnetoplasmonic nanoparticle that we made in our lab with polyethylene glycol to increase the biocompatibility, and we didn't see any long-term side effect for that, but there are some other options. We also used alginate to coat and also to incorporate gold and iron oxide nanoparticles. And as you know, alginate is another option to increase the biocompatibility of the complex that you are working on. So we have lots of options for that. And honestly, we didn't do a long-term study, let's say for a couple of months for the animal. By long-term, I mean at the most 3 months. So maybe after 6 months, you see some other side effects on liver or some other organs. But I personally didn't do this research, but I think you should find some other papers in the literature, because there are extensive and massive amount of research in this field, not specifically for gold coated nanoparticle, iron oxide nanoparticle, but for these two kind of nanoparticle. I'm sure there are lots of papers addressing biocompatibility of these nanoparticles. And regarding charge, I should say that what we were working on, all nanoparticles that we were working on were negative, but the ideal, you know, is to be positive, because internalization of positive particles into cancer cells and also in stem cells, this is the same for that. Uh, the ideal nanoparticle is positive nanoparticles, so maybe uh, chemist or physicist can do some modification to get the nanoparticles positive and increase the chance of easy internalization of nanoparticle into the cells. And the other question that you ask is size. Yes, size is very important parameters, and we have to take care of that. For imaging aspect, I have to say that it depends on the application that you are going to do. As I mentioned, if you are going to do T1 weighted MRI, you have to work with small, super small nanoparticles, let's say smaller than 5 nanometer. But if you are going to do T2 MRI, then you have to work with bigger nanoparticle, bigger than 20 nanometer. But when we look at physiological and biological aspects, there are some other things that we have to keep in mind. If nanoparticles are bigger uh, than what I mentioned right now, they all accumulate and trapped in liver, and so you cannot do any treatment or imaging. And so size is very important, and it depends on the application. But I know physicists and chemists, material engineers, it's that there are lots of options to them available to them that do fine tune in terms of size, and it depends on the application that you are going to do. Sometimes we are going to only look at liver, so it's fine if we use a bigger nanoparticle because the target is liver. But sometimes we are going to look at a lymph node. If all the nanoparticle that I inject into the animal or into the patient trapped in liver, there is no nanoparticle to be taken up in lymph nodes. So we have to do some modification in terms of size, in terms of coating layer or charge, as you mentioned. So you know, it totally depends on the application that we have in mind, and we can modify the size of the nanoparticle. And the last question, if I mention it here correctly, you asked about preparation. I have to mention that we didn't prepare these nanoparticles, so I'm not aware of the method of preparation. We have mentioned that in our paper, but you know, our colleagues from Department of Chemistry helped us to make this nanoparticle, and sometimes we buy the nanoparticle from companies. For example, in our MPI research, we buy Resovis, that is commercialized formulation for super SPO or for super paramagnetic nanoparticles. So it depends on the application, but we don't work on preparation, and it's always outsourced, and we have collaboration to get the nanoparticles. So I cannot answer your question about the preparation method, but this is my answer to your questions.
Thank you, sir. Thank you, sir.
Sure.
So, any other question? Okay, Mason.
Hello, Prof. Shakir. Thank you for your presentation. I just saw in your presentation some slides about the using of console software, console multiphysics for the thermal performance investigations and the other things probably. I just want to ask this, to what extent this perform these investigations just contribute to the impact of your paper? And the other question is that is it just used for only the heat transfer and I mean the normal model of heat transfer just used to investigate with this software or not? The question is just this.
Thank you for your question. Yes, the module that we used is heat transfer and also the optical modules, and also we built the geometry using the MRI, and in other paper we use CT as the actual and real geometry, and we import the CT and MRI images into console to make the geometry and then define the borders and boundaries and other things that you know better than me. And we use bioheat equation when you have a biological environment, in that you have blood circulation, so convention and every other biophysical parameter should be taken into account. So yeah, these are the modules that and equation that we used. But if you want to get more information, you can refer to our paper that I listed here, and also you can Google it and look at the details that we reported and explained in that paper. But about the first question, yes, if you use console simulation or any other simulation, the first benefit is that you will decrease the number of animals that you are going to use for increasing the therapeutic ratio. You can do lots of simulation without killing any mouse for your research, right? So this is the first important thing that you will show with your publication. If you before any in vivo and animal study, if you develop a computational modeling method that is verified, that should be verified at first, that is verified, and using that modeling, you demonstrate that this is the best and fine-tuned and optimized method for treatment. Absolutely, it will increase the impact of your paper, because you have everything in your paper in terms of modeling and then in vivo studies. So this is the answer to your first question.
All right, thank you so much.
Sure.
Thank you, Mason, for your question. Any other question? Otherwise, thanks again, Ali, for your presentation. We really appreciate your time to give us this presentation. So we will work further together to initiate some collaboration.
It's my pleasure. Thank you.
Yeah, thank you. So I'm just saying that next month, November, we will have we will run our next webinar. That the title and details will be announced later at UNISA and Itemba Labs platforms, or even we will send it through emails to all participants that they joined us today. So that was for our webinar today. Thank you very much, everyone, to attend. Thanks, Ali, for your presentation, and have a good day.
Mahmoud, please, if you just allow me just to mention that we will be talking between us, and we will be liaising with Prof. Shakerizada accordingly soon. Thank you.
Sure.
Thank you so much.
Yeah, sure. Thank you. Bye-bye. Bye, everyone.
Have a good day. Bye-bye. Bye-bye.
Bye.